Western blot, or immunoblotting, is a research technique used to identify protein, measure its expression level, and confirm the presence or absence of a protein of interest. The entire process requires 9 steps. Step 1. Protein isolation and purification. We begin with cell lysis to release proteins from samples. After selecting the sample from tissue, cell, or other protein sources, we extract proteins by making pores in the cell membrane so proteins can come out. There are 2 ways to extract: mechanical disruption or by the action of chemical reagents. Chemical reagents are more efficient at extracting internal proteins. For Western blot, we use ionic detergents and denatured lysis to unfold protein structures into a linear form. We also add protease inhibitors to prevent enzymes from digesting proteins. Optionally, we use a sonicator to further break open cells. Now we have extracted proteins. In the next step, we check the quality and quantity of the protein to ensure that we have enough to continue. Step 2. Calculating protein concentration using BCA assay. Prepare a 96-well plate. Create a protein standard using bovine serum albumin (BSA) at known concentrations. Load the protein extracts with unknown concentration and BSA standards in the wells. Then add reagent containing copper-2 ions. When BCA binds to protein, it reduces copper-2 to copper-1 using Burette reaction. Copper-1 interacts with BCA to form a purple color that absorbs light at 562 nanometers. This is called colorimetric detection. Then, we incubate the plate. The color intensity changes depending on protein concentration. Use a spectrophotometer or microplate reader to measure absorbance. From the results, create a standard curve and calculate the equation on Excel. If R square is close to 1, we can continue. Sample preparation. Once the protein concentration is known, mix the protein with sample buffer containing SDS, glycerol, to mercaptoethanol, bromophenol blue, and tris buffer. SDS gives the protein a negative charge and denatures it into linear form. Glycerol increases sample density so it sinks into the gel well. To mercaptoethanol breaks disulfide bonds. Bromophenol blue is the tracking dye. Tris buffer maintains pH. Heat the sample to fully denature the protein. Step 4. Gel electrophoresis. In this step, different proteins are separated by size and charge. Prepare a polyacrylamide gel. Use timid to help it polymerize. The gel has 2 layers: stacking gel on top and resolving gel in the bottom. Stacking gel has large pores to line up proteins, and resolving gel has smaller pores to separate proteins by size. There are 3 differences between the layers: the pore size, polyacrylamide concentration, and pH. Load samples and run electrophoresis. SDS gives protein a negative charge, so they move towards the positive electrode, which is on the bottom. Smaller proteins move faster and go further down, and larger proteins stay closer to the top. Bromophenol blue tracks their movement. Step 5. Blotting to polyvinyl iodine fluoride membrane, or PVDF membrane. In this step, the separated proteins are transferred from gel onto a PVDF membrane. There are several reasons why we do that. Gels are fragile and not suitable for detection. Antibodies cannot bind well inside gels, and PVDF membranes are durable, flat, and good at binding proteins, making them ideal for detection. First, we activate the PVDF membrane by soaking in methanol to make it hydrophilic. This improves protein binding and allows antibody solution to spread evenly. Then, soak in transfer buffer with methanol and stack the layers. The stacking is done in the following manner. From top to bottom, sponge, filter paper, PVDF membrane, gel, filter paper, and sponge again. This stack is placed inside a transfer cassette in a tank with buffer and electrodes. Proteins move towards the positive electrode. The PVDF membrane is placed on the positive side, so proteins move from gel onto membrane. Optionally, stain the membrane with Bonacieux S to confirm that proteins transfer successfully. Step 6. Non-specific blocking. Before adding antibodies, we soak the membrane in a blocking buffer. The blocking buffer is usually made of non-specific protein blockers like 5% milk or BSA. If we do not block, antibodies might stick randomly, causing background noise or false signals. Blocking buffer fills in blank spaces on the membrane so that only specific antibody binding can take place. Step 7. Primary antibody incubation. In this step, we add the primary antibody, which is designed to bind only to the target protein. After adding the primary antibody, we incubate. After incubation, we wash the membrane using buffer with twin detergent like TBST or PBST to remove unbound antibodies, reducing background. Step 8. Secondary antibody incubation. In this step, we incubate with a secondary antibody which binds to the primary antibody and is conjugated with a chemical reporter, like HRP. The secondary antibody has two main jobs. One, it makes the signal stronger. Since many secondary antibodies can stick to one primary antibody, it helps the signal appear more pronounced. Two, it helps us see the protein because it carries a special enzyme that produces light when we add a chemical. This is called chemiluminescence detection. The principle behind chemiluminescence detection is that when secondary antibody reacts with the substrate, it produces light, which can be detected as signals. Step 9. Signal detection and data quantification. After secondary antibody incubation and washing, we detect light emission using a digital imaging system. Then we interpret data from the image and compare with housekeeping proteins like actin and GAPDH. We try to determine presence or absence and expression level of the protein of interest and analyze differences between experimental groups.